Hello, all you happy people. I'm sorry that I was away for such a long time. But, do you remember this one? But I have to admit, I'm not really keen to debunk his new video yet again. But I decided to do it anyway. Why? Because I learned a shitload of things in past 2 years. Mostly about biology, biochemistry, genomics, genetics, evolution in general, and of course, video making. So, Mr. Doesitaddup101, Mr. Doesitaddup101, are you ready?
HAR1 is a region on chromosome 20, specifically the long arm of chromosome 20. It's 118 base pairs long.
Wait, am I seeing and hearing this right? You are doing actual research? No, that's not possible. You don't do research, you just do medical stuff thingy, right?
I have a background in both in research science and in medical science. From a personal experience, I believe medical science is more evidence-based.
Yes, that just means that you owned yourself in 30 seconds flat. You win. I could just stop the video right now and that's it, but nah, I just want to fuck with your head just a little bit more.
So what is special about HAR1? The rate at which DNA changes can be plotted on a bell-shaped curve.
Oh, isn't that cute? You just owned yourself with the fallacy you pointed out yourself. Oh, you don't know? Let me refresh your memory.
Proof by verbosity. That is, an attempt to con the readers of the site into accepting their arguments by presenting so many studies that the reader is overwhelmed with seemingly reliable information, thus blindly accepts their arguments of the authors without checking further.
There we go. Proof by verbosity. You're trying to repeat the argument that you lost the first time around and convince the audience that the science is on your side. The first time it wasn't, the second time it still isn't. Try again.
Most regions of DNA change at 0.01 substitutions per site per million years. This is the average rate of change of DNA.
Well, no, not really. First of all, that number might be only valid in genetic equilibrium. The other one is that this number varies a lot with the size of the genome and size of the site itself. Also, in my previous video addressing this exact issue, I've shown research that basically avoids that number.
A lot of species have this region on chromosome 20. For example, chimpanzees, chickens, foxes, and rabbits. When we compare the chicken and chimpanzee version of this gene, there are only two differences. When we compare the chimpanzee and rabbit version of the gene, there is only one difference. When we compare the fox and chimpanzee version of the gene, there are actually two differences. However, when we compare the human and chimpanzee version of HAR1, there are actually 18 differences.
Holy shit, somebody open Scientific American! You never bothered to read the article in full, did you? Or did you just stop at that HAR1 stuff thingy? Hello? Hello? Anyone there?
This is very surprising because the common ancestor of chickens and chimpanzees is thought to have lived more than 150 million years ago, while the common ancestor of humans and chimpanzees lived only 6 million years ago. How could 18 substitutions occur in only 6 million years between humans and chimpanzees, while only 2 substitutions between chickens and chimpanzees?
So wait, your problem is that there weren't enough random mutation in an extremely small region of DNA, namely 118 base pairs. So what?
So what is the molecular function of HAR1?
Oh, so you're going for molecular function of HAR1 gene. This is going to be very interesting. I hope you're not ignoring other components. You know, like usual.
HAR1 produces an RNA called HAR1F. HAR1F controls the function of a protein called relin.
Okay, where's the rest? Come on, I know you have it somewhere. I'm kinda disappointed. I was hoping that you're actually going to tell us about renin. You know, the protein that's needed for development of the thing called neocortex. The one that's not only dependent on HAR1F RNA. You know, the one that's also dependent on DAP1 gene and renin gene. I'm not really sure of which version of renin you're talking about, but this is the version of renin that I can find in, scientific literature.
So this is very exciting because HAR1 is at least one of the genes which is likely involved in producing the chimpanzee and human differences in the neocortex.
So what exactly is the point of this video? You're trying to address one region only, but you also acknowledge that there might be other things that influence the development of neocortex. So this video is void? I give up. I have to admit that I'm not quite sure what you're trying to prove. Your inability to decide? Inability to read scientific paper? What?
So how did this region change from the chimpanzee version to its current human version?
Humans. Chimps. Humans are not descendant from chimps, you fucking idiot.
This is actually the most crucial part of this video. The long-running debate in evolutionary theory for the past 90 years is whether most changes to DNA occur as a result of natural selection or simply random changes.
Emphasis on one tiny little word. Most, not all. Also, your further explanation of...
So the two branches of modern synthesis are the so-called adaptation model and the neutral model.
Makes absolutely no fucking sense whatsoever. You're acting as these two models are completely exclusive from one another. Well, no they aren't.
Some of the mechanisms of the neutral model are genetic drift.
Here, let me help you fixing this tiny little word. Oh, and psst, it's biased gene conversion. It's not biased genetic conversion. Just for informational purposes.
Population bottlenecks and biased genetic conversion.
Gene conversion. I also love how you describe in detail population bottlenecks and biased gene conversion, but completely ignore genetic drift. You mentioned it in the beginning, yes, but after that, no more. Now, why am I mentioning genetic drift? In previous video, Mr. Doesitaddup101 brought up a paper called Adaptation or Biased Gene Conversion, extending the null hypothesis of molecular evolution. But since he is also referencing in this video, I thought I might brought it up. I would also like to point out that co-author of that paper, Laurent Duret, wrote an article on nature.com called Natural Theory, the Null Hypothesis of Molecular Evolution. And it's not ignoring genetic drift, but heavily relies on it. So one of the things that Mr. Doesitaddup101 didn't manage to read? Wouldn't be the first time. The rest of the video is mostly pointless garbage, while it does explain how things work. Except applying that knowledge takes a brain cell too.
In this study, it was concluded that strong biased genetic conversion counteracts purifying natural selection and leads to the fixation of harmful amino acid mutations. The researchers termed biased genetic conversion the Achilles heel of our genome.
Thank you very much for showing us that you're completely incompetent to read the title, let alone everything else. Ladies and gentlemen, Mr. Doesitaddup101. Also, this paper concludes, and I quote, concluding remarks. This work demonstrates that GC biased gene conversion, like adapted evolution, can lead to strong and sudden accelerated evolution of functional elements and impact D and DS ratio, thus potentially corrupting positive selection detection tests. Therefore, one must be cautious when interpreting fast evolution in adaptive terms. Selective hypotheses should be invoked only after both the neutral and the GC biased gene conversion models have been rejected, the extended null hypothesis of molecular evolution. The rest of the video is nothing but garbage. I mean, not like the beginning was anything better, but still. Oh, and next time, when you try to decide how good and awesome and smart you are, at least listen to the ones that are smarter than you. Just a thought. I also love the fact that you removed this from your channel. This channel was meant for intelligent atheists, agnostic, and Christians who want to debate their beliefs in a rational and civilized manner. But whatever happens, don't be smarter than me. Don't learn about biology. Just watch my channel. I'm awesome. What intellectual discussion. I have something or other from something or other science, right? Also, I have no idea how to tell you that, but you kinda suck at research. Also, watching throughout his video, I found much better and fun hobby. Watching the paint dry. Bye bye.